Hello, ladies and gentlemen. This is Dan Montano, Chief Executive Officer of Zhittya Genesis Medicine. Today, we're having our webinar on Alzheimer's disease. Dr. Jack Jacobs will be giving the presentation, and he'll be starting in a few minutes. Before Jack gives his presentation, I want to do a little quick overview because I believe it's very important for all of you to understand that unlike some of our prior presentations, where we're talking about treating heart disease, and here is our evidence, or we're talking about diabetic foot ulcers, and here's our evidence, or we're talking about Parkinson's, and here is what we believe is the problem, and here is the evidence that we have. Alzheimer's is a disease which nobody knows really what causes it. There are different theories. One theory, beta amyloid plaque, has been pretty much disproven. And because of that now today, in Alzheimer's research, the question of what causes Alzheimer's is not agreed upon, and therefore it makes the question of how do you fix it more difficult. In the presentation that Dr. Jacobs is going to give, I'm going to ask each one of you to think of two things. One, is our hypothesis possible? Jack is going to explain what we and others believe is the cause of Parkinson's disease. We have not been able to prove it, but we have enough evidence that we think we may be right. But proving it, that this is the problem, is a big deal. Secondly, Jack is then going to go to the factor of if what we guess is the problem is lack of blood flow, then here's our idea on how we can remedy that and reverse Alzheimer's and bring people back. Now, why this is important is a whole bunch of medical reasons. First of all, there's something I learned a long time ago. Answers are very easy once you understand what the question is. Most people don't realize that having the correct question is often much harder than coming up with an answer. Now, Jack and I have a very good relationship. I guess and think and hope and say, Jack, could we do this? Jack, could we do that? Jack, could this be the cause? Jack, could this do that? And Jack says, your guesses are good, Dan, but I have to prove it to the regulators and to the scientific community, and his job is much tougher. Today, you're going to be our audience. You're going to be our judges. We're going to tell you our hypothesis of what we think causes Alzheimer's, and then Jack's going to present what we think is a way to treat it or remedy it. I want to make one comment why this is also important. In October of last year, Biogen, a large pharmaceutical company, was working on a treatment for Alzheimer's. Their clinical trial failed spectacularly. It was absolutely a disaster. A few months later, they came back in October and said, well, we might have a way that we could use it to treat Alzheimer's. In that one day that they announced their Alzheimer's drug may be viable, their stock market value as a company went up $15 billion in one day. The reason I mention this is that because there has not been any therapy for Alzheimer's for a long time, because the world is desperate for something, even Biogen taking a dead horse and saying it may not be dead, created a market value increase in one day of $15 billion. Now, I'm still simple enough to be impressed by making $1 billion or $15 billion in one day. When that doesn't impress me, folks, we're all going to be very happy about me. But the important question is, what is the cause of Alzheimer's? Do we have the right question? And then can we treat it? With this, I would like to introduce you to Dr. Jack Jacobs. Jack has been my partner for over 20 years. Jack has a PhD from the University of Washington in St. Louis, WashU. He is a PhD in molecular biology. He worked at Hitachi Pharmaceuticals. He worked at Merck, setting up their biological activities. Been with me for 20 years. He's one of the smartest people I've ever met, but folks, he is also the nicest person I've ever met. With this, I'd like to turn over to Dr. Jack Jacobs to introduce you to our theories and our thoughts on Alzheimer's disease. Dr. Jacobs.
Okay, thank you, Dan, for the nice introduction. And as Dan mentioned, we're going to be thinking a little bit outside the box, but not really that far. I think you'll be amazed how many people now consider blood perfusion, blood vessels, to be very important in the brain and for brain disorders. So I hope to convince you that we're onto something here. And as Dan said, there's desperately needed new medicines to treat Alzheimer's. Okay, so I'm going to first talk a little bit about blood vessels, angiogenesis, which is the formation of new blood vessels, and then move on, show you some data which we have gotten with our drug in an FDA clinical trial, and then move on into the brain disorders. We have about 60,000 miles of blood vessels in our bodies, enough almost to go around the world 3 times. And we know if you have blockages in vessels, say in your legs, I talked about this last week, you can develop peripheral artery disease, blockages in the heart, lead to heart attacks, angina. And then more recent data, blockages in the brain can lead to such things as stroke, Parkinson's disease, and we believe Alzheimer's disease. So let's take a look at blood vessels. They come in all shapes and sizes. They adapt to their location in the body. Here are 4 examples of different types of blood vessels. In the liver, the vessels are there to purify the blood, remove toxins. Here in the lungs, these are blood vessels now in the lungs, adapted to exchange gases, bring in oxygen to the blood and give out carbon dioxide. We'll be talking about vessels in the brains, feeding the billions and millions of nerves that are in our brains. And finally, here's in a contracting muscle. You need these curlicue type of vessels so they don't get kinked up as the muscle is contracting. We believe blood perfusion is critical. And in fact, over 75 human diseases have been shown to be a result of impaired blood perfusion. Heart disease, strokes, and as I'll talk about later on, neurodegenerative diseases are now thought to be initiated by a lack of blood flow in certain areas of the brain. So if you have a disease or disorder where you're missing blood flow, blood perfusion, the tissue becomes starved, ischemic is another word for it. What's a good way to treat that tissue? Well, one way is to grow new blood vessels, bring in new pipes. This is called angiogenesis, and it's a process that we use all the time in our bodies. Let me just give you an example of normal angiogenesis. Here we have a skateboarder, fell off his skateboard, scraped his elbow. This guy's going to use angiogenesis to heal that scrape. So let's look at that more closely. Let's say this is the wound on the skateboarder's elbow. Let's say there's a scab on his elbow. We're looking underneath it. And here you can see all these new blood vessels, tiny new blood vessels. This is angiogenesis in action. And this process is stimulated by the drug I'm going to be talking about, the drug we're developing. It's probably the most potent stimulator of angiogenesis in our body. So this is going to heal this wounded area, and it's going to regenerate not only skin, but everything that makes up skin, including nerves, blood vessels, hair follicles, sweat glands. So it really is a regenerative process, restoring skin back to normal. So let's look at the drug that we're developing and which stimulates this process. It's called FGF1. It's a protein, a biological protein. As I mentioned, it's a very potent stimulator of this angiogenesis. I actually was involved in purifying this at Washington University in St. Louis, where I was a graduate student. We were getting it from cow brains. And it turns out brain is probably the richest source of FGF1. So we know it must be doing something there to be there in such high quantities. Let's look at how FGF1 stimulates angiogenesis. Okay, so this is a blood vessel. And let's say you cut yourself, your body rapidly makes and releases FGF1. These little flying things are depictions of FGF1. coming to the wounded area, and they bind to the FGF receptor shown right here. So this is a very important interaction. FGF1, kind of the key, locking in with its receptor, and that starts a whole cascade of events. You get endothelial cell activation, and here you see those endothelial cells start to divide, and that's the beginning of a new blood vessel forming. As we see here, it forms a tube, and then here we get capillaries, new capillaries. So the thousands of capillaries you saw in that previous picture are all being stimulated by FGF1 here. Now, I'm often asked, what happens in the rest of the body? Why don't you get blood vessels formed in the rest of the body? And that's a very important concept. So here's FGF1, a key locking down on its receptor, stimulating all kinds of activities in the cell that lead to cell division and new blood vessel formation. So we want that only to happen in the wounded area or the ischemic tissue. And the mechanism for that is very important to understand the safety aspects of our drug. And that's shown here. Let's think of heart disease. Someone has a healthy heart, and over years or decades, they develop coronary artery disease. The tissue becomes ischemic, the heart muscle is starved because of lack of blood flow, less oxygen, less nutrients. Let's see what happens over time in that heart as it becomes ischemic. In the healthy heart, there's not much going on with the FGF system. Here's FGF1, here's a few receptors of FGF. Research has shown there are about 100 or so receptors in healthy heart, really not enough to stimulate new blood vessel formation because you don't need it. It's a healthy heart, you don't need enhanced perfusion. But over years, as one develops heart disease, researchers have shown now the number of receptors has gone up to 10,000 receptors. So basically, the heart is crying out for new blood vessels, new perfusion. And for whatever reason, there's not enough FGF1 there, so we come in with our drug and we stimulate it with what we call therapeutic angiogenesis to get that explosion of cell activity that leads to new blood vessel formation. Here's an artist kind of rendition of that in the starved or ischemic tissues. These are in the endothelial cells lining the blood vessels in the heart. Let's say the damaged heart, they know they need more blood vessels, basically crying out. And here we come in with therapeutic angiogenesis. This is FGF1 saturating those 10,000 receptors, and we get the explosion of new blood vessels in the heart. And I'm going to show you a video in a few seconds of results from a clinical trial where you can actually see those blood vessels forming in a person's heart. Now, we've used our drug in angiogenesis to treat, we're currently developing it for 19 out of the 75 medical indications where there's a lack of blood flow to a tissue or an organ. In previous webinars, I've talked about using it to treat diabetic foot ulcers. Last week, peripheral artery disease. Talked about coronary artery disease. And we're now moving into neurodegenerative diseases. We've talked about Parkinson's, Alzheimer's today. Next week, Vika Montana will talk about multiple sclerosis. Before we venture into the brain, let me just show you a little bit of data from a clinical trial that I actually was very much involved with, supervising it, and got some very nice results with our drug. So in this FDA authorized clinical trials, FGF1 was directly injected into a person's heart that had severe, very severe coronary artery disease. And after about 3-month period of time, we went back and looked to see if there were new blood vessels growing, and indeed there were, and that will show up on this video, which I'm going to show you right now. So ABC News went down to our site in Cincinnati and interviewed the neurologist, cardiologist, Dr. Lynn Wagner, and some of the patients. And that's, we'll show you on this video. And look at the angiogram. Lynn Wagner is going to point to the person's heart, and you're going to see very dark blushwork of brand new blood vessels that grew in this person's heart after they were injected with FGF1.
But since receiving an experimental treatment for his blocked arteries, his pain is gone.
I really feel great.
Duke was one of the first heart patients in the country to be treated with a protein actually capable of growing brand new arteries. The genetically engineered protein is injected directly into the heart. Within days, a network of new vessels begins to grow around the blockage, increasing the blood supply. Dr. Lynn Wagner showed us the changes in one patient's heart.
We see a small, narrow main artery and not very many secondary and tertiary arteries. This is after the treatment. What we're now seeing is new blood vessels growing here off the end of this artery.
And the patients themselves?
Symptomatically, they're improved within a couple of weeks of the treatment.
Just ask Constance Donnelly.
Oh, I feel wonderful. I've never felt so good in the last 5 years.
But doctors already see potential in other cases where the blood supply needs a boost, such as strokes and diabetes.
So the woman we just saw, Constance Donnelly, she was amazing. She came in as a cardiac cripple. She was in a wheelchair before the trial. You can see 3 months after treatment, she was up and about. She probably responded the best to the treatment, and remarkably, we had 3 dosing groups in that trial. She was in the lowest dosing group, got what we thought was 1/10 of the optimal dose of FGF1. So clearly her FGF receptors were upregulated in her heart to respond. So we're heading into the brain now. Your brain is incredibly vascularized, probably the most vascularized organ in your body. We're going to talk more about the microvasculature. And this is where the blood vessels meet up with the nerve cells in the brain. So we're growing small capillaries and small arteries. We're not growing these huge arteries, but I think I can convince you that the microvascularization is where all the action is. So let's look at this concept of the Neurovascular Unit. This is where the blood supply in the brain meets up with our nerves. So we start with these large arteries and going down to the smaller arterioles and capillaries. Here's a capillary. This is a healthy capillary. The lumen is wide open. Blood can get through there. Here it can give nutrients, glucose. Actually, the brain uses about 1/3 of the glucose that we take in every day. So oxygen, and then very importantly, removing metabolic waste products from those nerves. We're going to see in both Parkinson's and Alzheimer's disease a buildup of toxic proteins we believe are not being taken away because of the inadequate blood supply. Also, we'll talk about it a little bit later on, but there are stem cells, immature cells, that when neurons get damaged, normally can give rise to new neurons, but with a lack of blood flow in these diseases, those stem cells are not nearly as active. Okay, so we know from work of others that angiogenesis is coupled to neurogenesis in the brain. And then this highly schematic rendition we can see here, if we grow new blood vessels in the brain with angiogenesis, neurogenesis, so the growth of new neurons, are coupled to that. And I'll just show you one example. We don't have data in an Alzheimer's model, but we do have data in a stroke model, an animal stroke model, which kind of shows this. So let me just show you this data quickly. We can give a rat a stroke. This is actually a human, but think of this as a rat. We tie off one of the arteries in the rat's brain, actually the middle cerebral artery, and that rat develops a stroke. So here's a stroke here. Area of dead tissue, and then an area of at-risk neurons. These are slowly dying off because they lack a blood supply. So we can treat these animals with FGF1 and see if we can reestablish blood flow in this area, and also can we save brain tissue. So let me show you. We actually gave a rat a stroke, treated half with FGF1, the other half got placebo, and then looked in their brains. So these are actually slices through a rat brain. I've said this before, but if you believe in reincarnation, you don't want to come back as a laboratory rat. So here's a rat. These all show the stroke volume. This white area is infarcted tissue, dead tissue. These animals are not happy. They can do some behavioral testing. They put them on a balance beam. They fall off the beam. We then treat those animals for 2 weeks with FGF1. And you can see the remarkable and significant decrease in the stroke volume here. Okay, these animals on behavioral testing do much better. And we actually can look in these brains and look at the vasculature, look at the microvasculature and what has happened. We know that they're getting better neuronally because they're performing better in the behavioral testing. So let's take a look at the vasculature, and it's pretty remarkable. These are the capillaries in a normal brain, normal rat brain, rich supply of capillaries. What's not shown there is these are supplying the millions of neurons, which would be in this area of the brain. We give those animals a stroke and we just decimate those capillaries. We then treat one group with FGF1, another group gets placebo, and after 14 days, you can see we've reestablished microvasculature in the brain in that area where the animal had a stroke. We're not quite back to normal, but we're certainly better than placebo-treated animals. These animals desperately try to regrow blood vessels over the 2-week period because the brain knows it's in trouble, knows it needs blood perfusion to regenerate neurons, but just can't do it very well. This is disordered, tangled. This area can even hemorrhage and bleed and cause more damage in the rat's brain. So we believe the same thing is happening in Parkinson's disease and Alzheimer's disease, and that a molecule such as FGF1, which can reestablish first blood flow and then new neurons form, makes sense to test in patients with both Parkinson's and Alzheimer's. Okay, so let me talk a little bit now about neurodegenerative diseases. Here's a healthy brain on the left, a person without advanced Alzheimer's. I mean, the brain just gets decimated. You can see Dan Montano calls this a grape and this a raisin, and I think it's clear there's dramatic loss of brain tissue, less neurons, less blood vessels, and these people have obviously have serious disease there. This is in American statistics. In 2014, 5 million Americans had Alzheimer's. By the year 2060, that is estimated to triple. 14 million Americans will have Alzheimer's disease in the year 2060 unless something can be done for that. Some more statistics. Every 66 seconds, someone in America develops Alzheimer's. Nearly 500,000 new cases of Alzheimer's in the US every year. This is something I wasn't really aware of, but almost 2/3 of Americans with Alzheimer's are women. Now let's look at the age dependence in Alzheimer's, showing 3 areas of the world: Americas, Europe, and Asia. So anytime someone gets dementia under the age of 65, it's considered early onset Alzheimer's or early onset dementia. But that's only about 10% of cases. 90% of cases occur 65 years of age or older. And you can see just the inexorable increase in Alzheimer's. I mean, look at this right here. 90-year-old population has over a 50% chance of having the disease. That's just a remarkable number. And there's no treatment for these people. As Dan mentioned, there have been no new pharmaceuticals approved over the last 20 years. There are maybe 3 or 4 FDA-approved drugs to treat mild to moderate cognitive impairment. I asked our neurologist who's working with us on this, how do they work? Do they work well? And he said, Jack, it's like giving the patients a cup of coffee. That's the kind of bump they get in their cognitive improvement. So clearly, new drugs are needed, and we think testing ones outside of the dogma, which Dan mentioned, here's where most of the drug development efforts have been going over the last 20 years. Normal brain, normal neuron, and then the 2 hallmarks in Alzheimer's disease are these beta amyloid plaques, as shown here. And actually inside the neurons, you get tangles. This is a different protein, tau protein it's called. For 20 years, they've been trying to get rid of these things with monoclonal antibodies, enzymes, anything to try to break up the plaques, and really nothing has worked. I'm going to show you some data not in Alzheimer's, but in Parkinson's, a primate model of Parkinson's, where we actually can get rid of the plaques that are seen in that disease. Now, a lot of imaging is done in Alzheimer's patients. This shows PET imaging. So this looks at metabolic activity in the normal brain. So red areas, high metabolic activity, mild cognitive impairment, less activity. And here's Alzheimer's disease, just a decimation of metabolic activity in the brain. So this has been, I'm going to talk now about an imaging study which was done, a very large imaging study that was done, which I think changed the world's perception and certainly our perception of a possible potential cause of Alzheimer's disease. Okay, this was the article published, a very prestigious journal, Nature. It came out of McGill University in Canada, in Montreal, but actually it was a large consortium of people contributing to the study. Over 100 different medical centers contributed imaging data. And they looked at the progression of Alzheimer's disease. So from healthy individuals to full-blown Alzheimer's and everything in between, basically covering a 30-year progression to Alzheimer's disease. And they're going to talk about the early role of vascular dysregulation. Okay, so keep that in mind. What does vascular dysregulation have to do with Alzheimer's disease? Okay, huge study. They followed over 1,000 patients with cognitive impairment. 7,700 brain images were looked at. And they looked at different abnormalities, beta amyloid deposition, the PET scanning glucose, structural properties in the brain. But for the very first time, they looked at blood flow in the brain. That is during the whole progression to Alzheimer's disease. Before this, it's remarkable that the authors noted before the study was done, the most highly cited study looking at Alzheimer's disease progression didn't even look at blood flow in the brain, completely ignored blood flow. So let me show you the results of this. This is a busy slide, but I want to just take some time because I think the results are very important. Okay, so here's the progression of Alzheimer's disease. Healthy controls, moving on to, this is mild cognitive impairment, moderate cognitive impairment, and here's full-blown late onset Alzheimer's disease, okay? This is full-blown dementia. So 1,000 patients, 1,100 patients were seen covering a 30-year progression span in Alzheimer's. And they looked at, like, here's memory. Green is memory. We know memory becomes abnormal as you progress through Alzheimer's disease. Red here is beta amyloid deposit. We know you get those deposits as you progress. But look at the very first symptom, the very first abnormality that pops up going from a healthy controlled into dementia is vascular dysfunction, okay? Preceding memory loss, preceding beta amyloid deposition. So our and other people's thoughts are, if you can flatten this curve, flatten this down, you perhaps could really slow down or maybe even halt the progression of Alzheimer's disease. Okay, and let's just look at this early region here from healthy control to when someone has this mild cognitive impairment. Again, this is loss of memory in these patients, beta amyloid, but the very first thing is vascular disruption. What these authors concluded, this is their paper, vascular dysregulation might be the earliest and strongest brain pathologic factor associated with late onset Alzheimer's disease development. So this is really, a new concept in thinking about how to treat Alzheimer's, to treat the vascular dysfunction first. And we don't have any data yet in Alzheimer's models, mainly because they were set up mainly to look at plaque. Let me just show you a little bit of data from Parkinson's disease, where, again, that's a neurodegenerative disease affecting a much smaller area of the brain than Parkinson's. But I think there's some really take-home lessons that are directly applicable to Alzheimer's from some of the work we've seen in Parkinson's disease. Let's look at blood perfusion defects in Parkinson's. Okay, so Parkinson's disease affects dopamine cells in the substantia nigra region. And this is a much smaller area, obviously, than Alzheimer's, but lack of blood flow, we believe, and I'll show you some blood perfusion data, lack of blood flow to this area causes the normal neurons to become dysfunctional. Less dopamine gets secreted, and you get all the movement disorders of Parkinson's, the gait disturbances, the tremors. If you look at perfusion, blood perfusion in this area, it's really remarkable. Using functional MRI, here's a healthy 21-year-old, maximal blood perfusion in that area where the dopamine cells are. As we age, less blood flow in our brains as we age, but not enough to kick off the symptoms of Parkinson's. And look at this here in Parkinson's disease. 50% reduction in blood flow, we think, is choking off the blood supply to those dopamine cells, causing Parkinson's disease. So we think the same type of activity could also be present in the initiation of Alzheimer's disease, certainly as that paper showed. Also in Parkinson's, other areas of the brain can be affected. Dementia, as one sees in Alzheimer's, is also common in Parkinson's. And that's going to affect the frontal cortex area of the brain, so where the hippocampus is and body awareness. Let's look at blood flow in a woman with dementia. Okay, here's another functional MRI scan of a woman with dementia. We're looking at the front part of her brain. Blue is normal blood flow. Yellow is reduced blood flow. Red is severely reduced blood flow in those areas of the brain which are responsible for cognition, executive functioning, memory. So we would predict if we could flood the brain with FGF1, we could increase new blood vessel formation and perfusion in these areas of the brain. I wanted to show you just a little bit of data from a monkey model of Parkinson's, because again, I think this is directly relevant to Alzheimer's. We can give a monkey Parkinson's disease with a toxin. We then treat them with FGF1 over a period of 7 months. Their motor skills get better. I'm not going to show you that data, but what I'm going to show you is when we look inside the brains of these animals, we see two things. So this is the area, the substantia nigra region, where the dopamine cells are. Look at all the new dopamine cells that FGF1 has regenerated in these monkeys, okay? So these monkeys are doing much, much better in their motor skills. Without treatment, no neurons are being formed. So we're attacking the root cause of Parkinson's disease in these monkeys, regrowing new dopamine cells. And we see no reason why that would not also be the case if we tried this in Parkinson's, in Alzheimer's disease. Now, equally important, we talked about the beta amyloid plaque in Alzheimer's. In Parkinson's, in both people and these monkeys, you get the same sort of plaque. It's made up of a different protein, synuclein, but still there. These are toxic to the neurons, to the dopamine neurons, just as the beta amyloid plaques are toxic to the neurons in the brain. After treatment with FGF1, you can see we sweep away a lot of this toxic material. Not quite back to normal, but certainly better than this situation. We believe regenerating the vasculature helps flush away these toxic tangles, and these two activities, getting rid of these toxic tangles, regrowing new neurons, cause the halt and progression of Parkinson's disease in those animals. So we're excited to try the same treatment for Alzheimer's disease. So we're going to submit for a phase 1 clinical trial in both the US and Mexico. We're going to treat patients with mild, at first, with mild to moderate cognitive impairment. We'll study 3 ascending doses, no placebo group. We're going to treat these patients a bit longer than those that have Parkinson's. We're going to do 1-hour infusions 3 days a week for 12 weeks. And we'll study the same kind of markers that the McGill study, look at memory, look at blood flow, imaging of cerebral blood flow, and follow the patients out for a year. So we're very excited to try this new approach to treating Alzheimer's disease with a emphasis on trying to recreate the microvasculature in these patients. Work from neurogroups have also implicated microvascular dysfunction in all these brain disorders, Parkinson's, ALS. We've recently got approval to start an ALS trial in Mexico. Next week, you'll hear about multiple sclerosis, Alzheimer's, chronic stroke, traumatic brain injury, PTSD, something the veterans and military are very concerned about. MSA is kind of an aggressive form of Parkinson's disease. In 2 weeks, I'll be talking about chronic depression, vascular dementia, and then CTE we'll be talking about later. This is this disorder that boxers and NFL football players get after repeated concussions and head injuries. Okay, I want to just end with a study from a very prestigious university, Columbia University Medical School in New York City, published a study in Cell, prestigious journal, and why our brains perform worse as we age. And the reason I'm going to show you this is that it really does support our hypothesis about blood flow. We believe it's very important for these neurodegenerative diseases. This study showed that it's also important as we age, that the reason we forget names and forget where our car keys are is that we have less blood flow to the brain. And I'll give you kind of the punchline. What they're going to show is that you need adequate blood supply and perfusion to get our stem cells to divide and form new neurons. So in younger people and in older people, we both have these pools of stem cells, but in younger people, they have a better blood flow to the brain. They're better able to replace neurons that need replacing in the younger individuals. This is the Columbia study. Why our brains perform worse as we age. Healthy people who died suddenly in accidents. We agree completely with that. Columbia researchers feel it's important in aging blood flow. We obviously believe it's critically important for these neurodegenerative diseases. I just want to end with something which is also very important, how we make our FGF1 for our upcoming clinical trials. This would not at all be possible without the use of genetic engineering techniques, recombinant DNA technology. FGF1 was first purified from cow brains. I mentioned can't use cow FGF1 for clinical trials. And there's really not enough FGF1 in the human body to really try to purify from humans. So without these bioengineering technology, we would not be able to really use FGF1 in clinical trials or hopefully as an approved drug. Using bioengineering, we put the FGF1 drug into bacteria. This is a manufacturing plant where we make the FGF1. The bacteria divide every 30 minutes. So in these fermenters here, you can grow up large quantities of bacteria which are making our human FGF1 protein. And I just want to show you a gel. I love these gels. I run thousands of them throughout my career. It's a protein gel, so we can take some of the bacteria out of that fermenter, crack it open, and run the proteins. Each one of these little bands is a protein. They migrate through the gel based on their size. If you apply an electrical current, here's the big proteins, medium-sized proteins, and then the smaller proteins. And here is our human FGF1 being made in the bacteria. So we force this bacteria to make FGF1 as the predominant protein in that cell. Bacteria don't need FGF1, they don't particularly want it, but there it is. And we then have to get rid of all these other bacterial proteins that are toxic or could create an immune response. And over weeks and different purification techniques, we wind up here with pharmaceutical grade, commercial grade FGF1, which is ready to go into people. So I'm happy now to answer any questions. And after the Q&A session, Dan Montana will come back for some concluding remarks.
Thank you, Dr. Jacobs. We have had many questions come in during the presentation. Unfortunately, we only have time for a few of them. Now, dear attendees, if your question was not answered, please email it to dan@zhittyamedicine.com and we will answer it as soon as we can. Now, Dr. Jacobs, for your first question, with the right to try law rule, why can't people use Zhittya to solve some of these problems ahead of approval?
Yeah, well, that's a good question. And there are these new right to try rules, and we would be open to that. The rules are that you have to first be in a US phase 1 clinical trial, and that people who cannot get into that trial for whatever reason can ask us, and with the FDA's permission, we can make the drug available on what's called compassionate use. And I think we're certainly open to that, especially for something like Alzheimer's disease. So once we get going in the US, I'm sure that'll come up, and we will consider requests, very seriously.
Thank you, Dr. Jacobs. The next question is, my father has had Alzheimer's for 10 years now. Would he be a possible candidate for a trial, or would this treatment work after approval for my father?
So as I mentioned, at first we're going to start with mild to moderate cognitive impairment. See how that works first, try to stop the progression. But my goodness, now we will try it later on in later clinical trials in more advanced Alzheimer's to see if we can even reverse it. And certainly once it's approved, neurologists can really use it in any patient they seem fit.
Thank you. For the next question, how are you planning on measuring the outcome?
So the outcome will be using standard questionnaires to test memory, cognition, and then we'll be having these exploratory imaging endpoints to look at blood perfusion. But the FDA really does want you to look at clinical outcomes, and that is to stop the loss of cognitive impairment.
Thank you. For the next question we have here, can this treatment be preventative?
That is a very good question. If, like the Columbia video showed, if it can prevent, if the aging brain and the cognitive decline with the aging brain is due to lack of blood flow, then perhaps people would take it to, keep their brain healthy, keep it younger. And in that sense, that would be preventative for cognitive impairment and perhaps the dementia of Alzheimer's.
Thank you. Now for our last question, would these patients need to go through rehabilitation like post-stroke patients after the treatment?
Most definitely, yes. That's a very good question. We can regrow blood vessels and hopefully regrow neurons and get these people back, but they're going to have to be rehabilitated, retrained. Memories would have to come back. And so, yeah, there would be a long period, hopefully not too long, but a period of rehabilitation after they receive the treatment.
Okay, that is all the time that we have now for questions. We'll be moving the presentation along to Mr. Dan Montana.
Hello, ladies and gentlemen. Zhittya is a Las Vegas-based biotechnology company. We are developing a biological drug that stimulates therapeutic angiogenesis. As Jack mentioned earlier, lack of blood flow to a tissue or organ is responsible for 75 diseases. And as Jack stated, we're working on 19 of them. And those 19, I believe, are responsible for the death and suffering of 50% of all the people over the age of 55 on this planet. So we believe it's a big deal. Zhittya's business is discovery. We have to discover how our drug works. What are the medical indications it may be applied to? what dosing, where is it safe, where do we have create an adverse event. Even chocolate, you can overdose on chocolate, you can overdose on salt, you can overdose on water. Everything becomes toxic if the overdose. What we have to do is find that sweet range where we have enough medicine to get an outcome that is positive without crossing the line into something that is more dangerous. We have to design and carry out clinical trials to demonstrate the drug is safe and effective in humans and that are caused by a lack of blood flow. Now, I want to take a moment here, and one of the questions that was asked is how are we going to measure? And Jack talked very much about the medical indications the FDA would want, and that being the clinical analysis on the people's memory. In Mexico, where we have permission to do the Parkinson's trial, one of the things that Jack's been talking to them about is they have a fantastic ability to image these cerebral blood flows. And part of what our protocol there is we're going to take a picture of people's brains before we dose them. And after we're done dosing them, let a couple of weeks go by because it takes about 6 weeks to grow the blood vessels, then take a picture and see if the area with a lack of blood diminished. If the area that was bad is this big and after our work it's this big, I believe we are on to something. And the clinical ability to measure if somebody can remember things or ceases to lose their memory may take us another 6 months or a year. But shrinking the drought area from this to me is a huge deal. And that's one of the things that I'm really proud that Jack has been able to get into the Mexican clinical trial on Alzheimer's. And we have to interact with government regulatory authorities to obtain approvals. And this is one of the other things that Jack mentioned. We're working with the US FDA. We're doing everything we can. They are not the swiftest organization. And this year, they've been taken to work on COVID-19. They basically stopped all clinical trials that were not COVID-19 related. So it's been very difficult to move on other things, such as what we're doing. Mexico's been the same, Estonia's been the same, and some other countries have. But we're learning, because we are very interested in not wasting time, that some of these countries, it is the hospitals that approve the drugs for use in those countries. And those are of particular interest to us because they've indicated they can move very fast. So that's also interacting with government regulatory authorities. If you want information on what we're doing, we've created a small booklet, and this is a copy of the cover: treatment for heart disease, strokes recovery, Parkinson's disease, multiple sclerosis, Alzheimer's, diabetic foot ulcers, etc. Just email me, dan@zhittya.com, and I'll send you a copy. This is a paper that Dr. Jacobs did, stroke, dementia, Alzheimer's, vascular disorders of the brain. If you want a copy of this, once again, just email me and we'll send this to you. Once again, the more you know, the better off we all are. A week from today, on September 15th, my wife, Vika, will be giving a presentation on multiple sclerosis. Is therapeutic angiogenesis a treatment or even a possible cure? And this talks about the 2.5 million people that suffer from MS. 3 out of 4 are women. There's going to be a lot of interesting things come from this webinar. But one of the things that I would like all of you who are following our webinar series as much to enhance your medical knowledge and your own health concerns is Jack mentioned to you, whether it's the Columbia School of Medicine or McGill or US University of Southern California, the brain shrinks as you get older. And the question is why? And one of the theories that's evolved over the last couple of years is endothelial cell dysfunction. And endothelial cells are the cells that make up the vessels. Your blood vessels are made up of living cells that stick together and work together to form a channel to move blood from point A to point B. And those living cells have to be fed, nurtured, oxygenated, waste removed, just like any other part of your body. And it is my belief, and once again, this comes back to my opening statement, it's easier for me to guess than it is for Jack to prove. As you may note, I can guess in 24 minutes. Jack could take 24 years to prove what I guessed is right or wrong. But the point being is, Vika will be presenting in the multiple sclerosis, the endothelial cell dysfunction that we've been able to uncover and understand to a degree. And to me, I believe it may be the fundamental underlying cause of all these neurodegenerative diseases. So whether it's Parkinson's or Alzheimer's, I believe the MS presentation is very valuable for you to see. After that, on September 22, Dr. Jacobs will be talking about chronic depression. Is therapeutic angiogenesis a breakthrough treatment? 16 million Americans suffer from one major depression episode per year. I will challenge that number. I think from March of this year till July of this year with this COVID-19, I think there was a lot more depression. I can tell you, I think it's just one of those things that happened. Now, this is something that Jack's been pushing me on for 20 years. He's a true believer that this can, FGF1 can make a difference in the condition of these people. So once again, that's going to be on September 22nd. Our program of outreach is very simple to make people aware of our science. And I think at least your mind is being expanded, you're learning. And two is to develop a network of motivated and informed individuals. We presently have over 40,000, it's probably closer to 50,000 now. And over the next 60 days, we'll be reaching out to over 1 million people. And I shouldn't say that, we've already started the 60 days, we've already touched 800,000. So we are rapidly trying to let people know what we're doing. If you have any questions, please feel free to email me at dan@zhittyamedicine.com. Visit our website, zgm.care. We appreciate your time. We respect the fact that you gave us your time to listen to what we're doing. I believe we have something big. I believe that 100 years from now, people will look back, or 100 years in the future, people will look back and see at this moment in time, the discovery of therapeutic angiogenesis to treat the diseases that were killing half the people was a medical breakthrough. And you were there, folks. Thank you. Have a good night. God bless you all. Bye.